Hi folks, welcome to another edition of Zog Science. Today we're going to be talking about polymers, monomers, how do we make organic molecules, and then we're going to get into carbohydrates and lipids. So macromolecules or organic molecules are polymers, and they are built from monomers. So a polymer is a long molecule consisting of many similar building blocks. Those little building blocks are called monomers. Some molecules that serve as monomers also have other functions on their own, so they're not necessarily just building blocks. They can have their own function. Cells are making and breaking down polymers all the time, and they do it via the same mechanism. A dehydration reaction occurs when two monomers bond together through the loss of a water molecule, sometimes called a dehydration synthesis reaction. And then polymers are disassembled to monomers by hydrolysis, where you reverse the process of a dehydration reaction. And both of these are facilitated by enzymes, which help to speed up chemical reactions. So in a dehydration synthesis, we're going to take a hydrogen and a hydroxyl group, or a hydroxide group, and they're going to be pulled out, and it's going to create water. So dehydration, you're taking water out of the monomer and polymer in order to link them together. And then in hydrolysis, you're adding in the water, and you're using that to break it down. So lysis means to break, hydro is water, so you're adding in the water in order to break the polymer apart. Now, carbohydrates are one of the main macromolecules that is found inside living things. They include sugars and polymers of sugars, and the simplest carbohydrates are called monosaccharides, or simple sugars, and the macromolecules are polysaccharides, and these are polymers composed of many sugar building blocks. Monosaccharides have molecular formulas that are usually multiples of CH₂O, so that's a 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. Glucose is the most common monosaccharide, and its formula is C6H12O6. And monosaccharides are classified by the number of carbons in the carbon skeleton and the placement of the carbonyl group, which is that COO. We are not going to be worrying about that too much. We're not going to get super into like needing to know how to name them or what all of the specific ones are. You should for sure know glucose though. These are just some different examples. There are trioses, which are 3 carbons. There are pentoses, which are 5 carbons, and there are hexoses, which are 6 carbons. Again, one of the things you'll notice is that a lot of them have the OSE ending, right? So that's a way to kind of know a sugar is if it has an OSE ending. Also, obviously, glyceraldehyde does not, so it's not necessarily a rule all the time, but many of them do. A disaccharide, di means 2, right, is when you have 2 monosaccharides that form together with what is known as a glycosidic linkage. So, for example, glucose and fructose, a dehydration reaction will occur, and they'll be put together, and that makes sucrose, right? And sucrose is table sugar. And so we've got this 1,2 glycosidic linkage between the two different monomers, creating the polymer. So polysaccharides, these are polymers of sugars, and they have storage and structural roles. And they store energy, and they also can help with structural things in terms of providing structure for living things and cells. The structure and function of the polysaccharides are determined by what monomers make it up, as well as the position of the glycosidic linkages, which we will take a look at. Example of a polysaccharide is starch. It is a storage polysaccharide in plants, right? So you get starch from eating, breads and potatoes, things like that, right? Those are plants. And so it's a way for them to store energy for a longer period of time than just the single monomers. And they store these in little granules. Most animals have enzymes that can hydrolyze the plant starches, so they make glucose available as a nutrient. So they're breaking down the starch in order to get at the glucose that is the monomer that makes it up. Glycogen is the storage polysaccharide in animals. So when we have excess glucose, we take that glucose and we store it as a glycogen, usually in the liver and also in the muscle cells, so that the muscles can access that energy quickly. If we take a look At starch, it's got just long chains of glucoses. Glycogen is similar, however, it is branched, so it's not necessarily in as straight of a line. It's got those branches that we mentioned in the last video. And then finally, there is also cellulose. Cellulose is another polymer that is made up of glucose. However, it has hydrogen bonds that occur between it, and it is a structural polysaccharide. It's not used to store energy. It is there to provide structure to the cell walls of plants. And let's go in and let's take a look at why is it that cellulose is different from starch in terms of its ability to act as a structure, whereas starch is more of an energy storage molecule. So the reason is that the glycosidic linkages in cellulose are different, and that's because it is based on two different isomers or enantiomers for glucose. So there's two different types of glucose, right? You can have alpha and beta glucose, and that all has to do with where this hydroxide group is located on the one carbon, right? So the enantiomers, it's again same chemical structure, but it has same chemical formula, but a different structure. And so because that hydroxide is located in a different location, it means that when we have alpha linkages between the glucose molecules as it is in starch, all of the hydroxides are all on the same side. And so those hydroxides can't line up with each other between glucose molecules of different chains in order to provide those hydrogen bonds. However, for the beta linkages of the cellulose molecule, the hydroxide groups are on opposite sides. And so because they are on opposite sides, they are able to provide those hydrogen bonds that allows them to hold together and allows it to provide a very strong structure. Now, the slight issue with this is that because cellulose has a different structure, right? It looks different. Enzymes, which if you'll recall from baby biology, are unable to break that down because it has a different structure. They can't bind to it. At least the enzymes that we have, right? So cellulose in human food passes through the digestive tract as insoluble fiber, and it's actually what helps to keep you regular, right? So the fiber, as it's going through your digestive tract, it scrapes against the linings of your digestive tract, which causes your intestines to release mucus, which helps to lubricate your intestines, which is the reason why you want to make sure that you're keeping a lot of fiber so you don't get stopped up. Some microbes use enzymes to digest cellulose, so there are microbes that can do this. And so many herbivores, including cows, right, or also termites, have a symbiotic relationship with these microbes. The microbes live in their gut. The microbes help to break down the cellulose, and then that provides energy for the herbivores. All right, so that's carbohydrates. We're going to talk about lipids now. Lipids are actually not technically polymers. They are not just a long chain of different things put together, but they have little or no affinity for water. And again, they're organic molecules and they're very important. They are hydrophobic because they consist of mostly hydrocarbons, which form nonpolar covalent bonds. So these are nonpolar molecules. They are hydrophobic. They don't want to be around water, which is their unifying kind of thing. It's why we lump them all together. One type of lipid are fats, and these are constructed from two types of smaller molecules, glycerols and fatty acids. Glycerols are 3-carbon alcohol groups with a hydroxyl group attached, so that's actually a polar section. However, the fatty acid is a very long carbon skeleton with just carbons and hydrogens, and so the fact that it's such a long nonpolar region basically balances that out. So here's the glycerol, right? Again, you can see how it is a polar portion. However, when that fatty acid binds, we've got our dehydration reaction. That is then going to provide such a long nonpolar region that it doesn't matter. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds. Unsaturated fatty acids have one or more double bonds. The reason why that's important is that your saturated fatty acids are solid at room temperature, which is usually your animal fats, and unsaturated ones are liquid at room temperature, which are mostly plant and fish fats, right? So examples are things like butter, right? Butter is a saturated fat. It comes from animals. And because they're all single bonds here, what that means is that these carbons and hydrogens are going to just form some nice straight chains, and so you can get all of the molecules lining up right next to each other, and so we are able to have that be a solid at room temperature. However, for unsaturated fats, because they have a double bond, that double bond results in a kink in the structure, and so that kink in the structure means that the molecules can't get as close to each other, and so it doesn't congeal into a solid at room temperature. It stays a liquid. The major function of fats is energy storage. There are some that are structural, but mostly it is for the fats, it is for energy storage. It's a way to have lots of energy stored for long periods of time. Another type of lipid is a phospholipid. This is two fatty acids and a phosphate group that are attached to a glycerol. The fatty acid tails are hydrophobic, but the phosphate group, right, we looked at that in the last video, it has those negative charges, and so it is very hydrophilic. It is good to be around water. It wants to be around water. And phospholipids are the major components of cell membranes because of this property. When you add phospholipids into water, they self-assemble, so they just spontaneously will self-assemble into a bilayer with the hydrophobic tails pointing to the interior and the hydrophilic tails heads pointing to the exterior. So this is that phosphate group again, along with a little choline section. Don't worry about knowing that, but it's hydrophilic, likes to be around water. Here is our fatty acid tails. And so you can see how we form this phospholipid bilayer with the hydrophilic heads on the outside, the hydrophobic tails on the inside. We'll talk a lot more about phospholipids when we get into our cells unit, and we'll talk about the lipid bilayer a lot. Finally, we've got steroids. Steroids are our final class of lipids. They're characterized by having carbon rings with 4 fused rings. Those rings can be different kind of setups, but one of them is cholesterol. Cholesterol is an important steroid. It's a component of animal cell membranes that helps to maintain fluidity. However, high levels of cholesterol are also potentially linked with arteriosclerosis. All right, thanks for watching, and I'll see you next time.